From the San Diego Convention Center in San Diego, California, this is the ASN Kidney Week 2018 podcast, a discussion of the latest scientific and clinical advances presented at this year's meeting.
Hello, my name is Mark Okusa. I'm the president of the American Society of Nephrology, and welcome to Kidney Week 2018, and also welcome to sunny San Diego. I'm pleased to be joined with three of my esteemed colleagues to discuss the events of the first day of our annual meeting in San Diego. So why don't you introduce yourselves and tell me a little bit about your background so we can take this into perspective. John?
Thanks, Dr. Okusa. I'm John Roberts. I'm an assistant professor at Duke University. So clinically, I'm a transplant nephrologist, and I spend most of my time and research time in medical education, sort of studying new and innovative ways to teach nephrology and physiology to medical students and residents. And I like to draw and make videos, so a lot of my stuff revolves around technology and using drawings and videos.
I'm Dinushika Mohottige. I'm a second-year nephrology fellow this year at Duke University. I actually came to nephrology and medicine via public health, so that's really, I think, informed why I fell in love with nephrology and wanted to practice nephrology clinically. It is all related to the fact that our field, CKD and transplant, is burgeoning with disparities, and so my clinical interests are in general nephrology, but research interests are in health inequities in CKD and transplant.
I'm Catherine Godson from University College Dublin, and I lead a research group. We have a particular interest in diabetic complications and in diabetic kidney disease in particular, and in inflammation. And my group has clinical fellows and basic scientists, so we're quite translational in our approach.
Terrific. So we have areas of interest that are quite diverse, from education to clinical nephrology to basic biology. So I think that's great. So I'd like to ask what your thoughts are about the meeting.
Well, if I can say, as having been to several ASNs, I think it is really inspirational how there's always something in it for everybody. So as I said, I have people from my lab that are here. So there's some clinical fellows, some PhD students, and then more established investigators. And there's really something for everybody. And I wasn't able to go to the basic research course because of other commitments, but one of the fellows from my lab went, and they were blown away, and particularly they really appreciated how there was diversity in the presenters. It wasn't only the very established big names, it was also more junior people, and they were really inspired by that. They sort of felt that there were clinical researchers doing basic research absolutely not at the periphery, so that they were really inspired. And then I thought that, if I may say, your presentation this morning was really fantastic. It was really motivational, and it was really interesting how it mirrored very much what came next about biochemistry being superseded by molecular biology, and then 100 years after the last advance in microscopy, this huge leap forward. So I don't know if you planned it that way, but it was really remarkable. I thought it was
yes,
great.
it was all planned.
Good. No, it was really
thank you for those
comments.
no,
it was good.
Yeah, I agree. In an unbiased comment, I mean, if you weren't here, we'd still be saying it was an amazing introduction and speech, and then Kidney X was announced, and then the state-of-the-art lecture was amazing. So it's kind of the thing, you had to be there to see the movies and the images that really popped, and just, it was amazing to see everything move on the screen.
But it's also, I thought, like really good example, role modeling for anybody getting going as a scientist or a clinician scientist as to how important collaboration is, how important basic science is, how, the technologies in a vacuum wouldn't have amounted to much. It was only when the people started to speak to each other, and when he was able to address, the kind of questions that we're more interested in. I thought it was really inspirational from that perspective.
Mm-hmm. Great. Other general thoughts?
I too thought your talk was so inspiring as a fellow and someone who's trying to get excited about the future in nephrology and what it holds for us. I think speaking to the past, present, and the future, and speaking directly to recruitment into the field and what's exciting and kind of what's on the horizon was a wonderful start to the day. I tried to go around to a range of different things, and from transplant to CKD related talks. I think mirroring your comments, the diversity, not only in the presenters, but in the topics. Everything from the bench to translational research has been represented already, and haven't even finished the entire day yet of seeing talks. So it's been fabulous as a fellow. Very, very exciting and career affirming.
Oh, it's great.
Well, thank you. I think those comments hit the nail right on the head in terms of where we're going from the past to the future. About the trainees, when you think about it, I've met a lot of STARS students who are here, and they are so excited to be here. And what's beautiful about that was they had 7 or 8 of them, plus they had a mentor there with them. And the mentors were people who were truly excited, people who actually are extremely busy. They were there mentoring them. They are very senior people who have this extraordinary passion to be with students and to ensure that they have a good experience here at the American Society of Nephrology Kidney Week. So I think this is really exciting. Is there anything in particular about any of the sessions that you might want to comment on?
Here, I have one question for the group. So at the State of the Art, or the plenary session, so with Dr. Betzig, he showed lattice light sheet microscopy. I had to write it down to make sure I retained it. Basically this amazing way to sort of visualize cells and molecules in motion. I just want to know from y'all, like, imagine you had like a day with him and his microscope, what part of the kidney you want to throw under the microscope, right? Because he showed neurons and what else did we see? We saw mouse cortex and we saw all sorts of Drosophila. So what about the kidney?
Maybe an SGLT2 treated kidney versus a control to see what really happens with glomerulus in response and high glucose.
It's really interesting. You know, he talked about bacteria, the brain, his high resolution, super resolution microscopy, and he talked about this one concept, one method called expansion microscopy.
Oh, with the
and
water.
What that is that you take tissue and you infuse it with certain material that everything is intact and the relationships between other molecules are intact, but it's blown up. And so everything is much more visible. And when I talked to him, he said, you know what, I didn't realize there were 850 million people in this world with kidney disease. I hadn't even thought about working with the kidney, but I need to. So I think that you're going to see Eric Betzig start working on the kidney, and I think that we'll have a better understanding of the molecules within single cells of the kidney and what they do. I find that very exciting.
And I was talking to a microscopist the other day who does very high-tech work as well, and they started explaining that, you know, as the basement membrane gets wider, how the cells that are supposed to be surrounding it just can't, they don't have the same trophic contacts. And he said it's what happens in the brain with astrocytes. And so I was just thinking, as he was speaking today, he could look at the same sort of things.
One of the things that I think is going to be exciting is the Kidney Precision Medicine Project. Whereas, and I mentioned this before, is that we tend to take kidney, we grind it up, and we get an average gene expression. But what this is going to do is allow for sequencing, single RNA sequencing within individual cells that allows us to really identify where the problem is, perhaps, and the location of where these problems are, so that we could really devise specific therapies. So what's interesting is that the work is going to be really unprecedented in the information that we get. So we start with Eric Betzig's single molecule, single cell imaging to Aviv Regev tomorrow in her state-of-the-art lecture on the Human Atlas, where we're going to understand more about single cells. And then we're going to move into translational work by Griff Rogers, the director of NIDEK, who's going to talk about his discovery and translation of hydroxyurea in sickle cell disease. And then we move on to population health with Rob Califf, who is the former commissioner of the FDA. So I think that they're all lined up to give us a broad perspective of the importance of different areas of medicine.
I really enjoyed the new diabetes drugs on the kidneys, the discussion of the mechanisms. I thought it was really interesting the way the incretin, targeting the incretin system, that had the DPP-4 inhibitors aren't anything like as effective as the GLP-1 receptor agonists. And there was so much dispute over, the last few years as to whether there were receptors, whether the GLP-1 receptor was in the kidney, and now seems to absolutely be there. And as the speaker was saying, it's sort of like reverse engineering from a really positive clinical trial to now try and work out the mechanism, which she's very much promoting. The idea that it was based on inflammation and that they were essentially anti-inflammatory. So I thought that was really interesting to go from actually the drug works to how does it work as opposed to the other way around.
Can you tell us a little bit more about incretin and its mechanism?
So, well, the way that the incretins stimulate insulin release. So and the GLP-1 agonists were developed, I would imagine, I hope I'm not incorrect, to supplement insulin release, but they found that they have these they show cardiovascular protection, which really wasn't expected, that seems to be independent of their glucose lowering. And so in the kidney, they also seem to be protective. So looking at albuminuria is protected, and then functional protection in terms of GFR. So it now would appear, we also know from the cardiovascular system that, for example, the GLP-1, the agonists, the different liraglutide is the one that I'd be most familiar with, that it, for example, can alter the macrophage phenotype, so from being an M1 to being more M2, which is, of course, a big simplification, but the idea now is that macrophages exist in very plastic states, and some are the classic ones you kind of would know about, the inflammatory, but others are more pro-repair. So there seems to be a dynamism between these states, and it seems that liraglutide impacts on those. So
And John, you're a renal physiologist, and what are your thoughts in terms of SGLT2 mechanism and renal function?
Yeah, it's obviously all the data is really exciting in terms of how it works, and obviously the mechanism is tantalizing in terms of thinking about our body has this really robust way to hold on to glucose, to hold on to energy. Like, we don't want to lose that, we want to conserve it, because we obviously adapted in an environment where we want to conserve every precious molecule we come into contact with. And now we have this environment where we may have excess of some of these things like glucose and carbohydrate and sodium. And so having this mechanism is really, really awesome. In terms of kidney function, obviously it's really exciting for all of us to see some of these outcomes in terms of slowing the progression of CKD and reducing albuminuria. And every time I bring this up, I get excited about using SGLT2 inhibitors in transplant specifically, and our pharmacists get they wince a little bit at the idea of a higher risk of urinary tract infections or pyelonephritis. But I get super excited about this idea of controlling diabetes in a way that doesn't require high doses of insulin, maybe 4 or 5 shots a day, because that's our default pathway, right? We default to high doses of insulin, and guess what? Our patients gain weight because we're treating their diet and we're treating the sugar rather than sort of treating some of the underlying physiology. So I love SGLT2 inhibitors in terms of the physiology.
Because they make sense.
Yeah, it makes sense. It just makes perfect sense. It's like, let's just block the reuptake of glucose and waste it in the urine. That makes so much sense to me. We're excreting the carbohydrate instead of forcing it into cells, and I think patients would enjoy that.
And also inhibiting RAS as well.
Totally.
Which seems to be the most intriguing aspect, isn't it?
Yeah.
So Dinushika, you're a second-year fellow. How do you find the ASN Kidney Week in terms of helping you in your career path?
I think that it's the reason why I got excited about nephrology in the first place is demonstrated here in essentially what all of you are doing, right? There are phenomenal educators who are all over this conference who, as you mentioned, are mentoring people actively, and I think the nephrology educators that I've been exposed to in medical school and through residency are so committed and devoted to teaching and so excited about what nephrology holds Being able to actually go on one of the ASN poster tours this morning through kind of a tour of AKI and EPI was incredibly exciting, and they 2-3 big leaders in the field in education were there guiding that tour. There were fellows and students on that. I thought that was fabulous. I think that sessions like the Best of CJSON and JSON is a fabulous thing for fellows, because you get kind of highlights of landmark trials, what is exciting, and from that new ideas are generated. I think that within each of the sessions, I actually went to the clinical transplantation, what everyone needs to know session, and it was broken up into immunosuppression and infectious disease and what are the benefits of the new allocation system. And so within that, in a 30-minute segment, a new fellow can see something really exciting, whether it's the advent of belatacept or the fact that disparities in race have been reduced with the new organ procurement allocation system. That is where I think the mind can start to churn as a new fellow, and as somebody trying to figure out what a research pathway might look like. Exposure to so much diversity in the presentations, and I think again the ongoing support of mentors and educators, the people who inspired people like myself to get into nephrology in the first place, is fabulous.
So Catherine, you're a part of the program committee.
Mm-hmm.
And it consists of both clinicians and scientists. How do you find the balance of programs, the balance of sessions with regards to the different areas of nephrology?
To be really honest, I find it very good in terms of how much basic science is integrated into that. There isn't such a huge divide, and I think it's really important that I think some learned societies take a very severe view of clinical and scientific and keep the two apart. But I think the strength of this meeting is that they're so well integrated. So for example, the new drugs for diabetes in the kidney, that was actually in the basic science session, even though it was predominantly around reporting from different trials, but it was really good. The perspective that was put on it, I think it's great.
There's one thing that I really enjoy about the meeting, besides all the quality science and the quality clinical and educational information, is the networking opportunity. It's amazing how you train with people for me a long time ago, and this is the one place we can get together and not only talk about what we're doing, but also to catch up on family and things of what's going on at their institution. And it's just a wonderful opportunity to do those things. Is there anything else that you all want to mention about Kidney Week, or anything that interests you?
Could it be a day longer? There's so many. You start at 7 in the morning and you end at 6, and you need to do the networking as well. But there's just so much on at the same time, and the conflict is always: is it to go to something in your area, or to actually go to something new? So it's kind of hard to balance everything, I think.
So one cool thing I just came out of was I was hanging out in the salt and water rooms, and Jens Tietz, he was giving his salt and water talk, and it's really entertaining. And the thing that brought this to mind, I was on a general medicine rotation a few weeks ago, and the residents were asking about all this related to salt and water balance, and I sent them his TED talk, and so maybe the listeners will want to look this up, but Jens has a TED talk done in Nashville, and it covers a lot of the stuff that he's presenting earlier today, but it was standing room only in there. I think next year you'll need a bigger venue, but it's just it sort of upends the classical teaching about salt and water balance, where our textbooks are correct, but only on day-to-day basis. So he had the opportunity to do studies with astronauts who were planning, a trip to Mars in Russia, and so they were doing this sort of simulated trip to Mars where they were locked in a metabolic cage for 500 days. And so it provided the opportunity for long-term balance studies with salt and water and weight and urine output. So it was really awesome. Basically, sort of everything you assume is not the case on a long term, and apparently we go into sort of 50-day cycles of retaining and storing sodium and then excreting sodium, and so it's really cool. I recommend watching that, and then also seeing him talk if you can find room to sit.
No, I heard his talk before,
Okay.
and I thought it was very interesting. And
It is.
I always wondered how the subjects participated in this and collected samples every day for I don't know how many days, but
Maybe they thought, hey, this is something to do to pass the time. You know, if you're locked in a biosphere, so to speak. Yeah.
So the other question I have is, you have been working with the app and the program book. What are your thoughts about that?
The app is great, although it needs to understand that you can go to more than one lot of posters in a 2-hour session, because it tells you, you know, you're already committed to 10 to 12 for this. But otherwise it's great. It's very easy to use as a reluctant apper.
We're saving paper at least, right? It used to be
We are.
the big book.
We would like to go green, so
It's great.
It's important.
I've
And it's also
loved
nice
the
to
app.
have this when you're in a real rush, you know.
Yeah, you're right. It gets upset when you try and click two things
Yeah.
at the same time.
Well, which is fine, but for posters they need to understand you can
Okay.
do more
Yeah,
than
yeah.
10.
And they usually we're hopping from one thing to another, like, ooh, I'm going to see the first half of this thing and the second half of that thing. But it's great. We love it.
We're
It's
good.
organized beautifully, very easy to use.
Yeah, isn't it?
So kudos to whoever made it for us.
Well, I tell you, I've been walking around and I've seen sessions that are overflowing and other sessions that were almost completely filled. So there seems to be a lot of excitement, a lot of buzz starting on day one. So I'm very excited that this is going well. So I want to thank all of you for joining us in this podcast, and I hope the rest of the meeting is exciting for you. Thank you.
Thank
Sure.
you
Thank
very
you.
much.
You have been listening to the ASN Kidney Week 2018 podcast. This podcast is copyrighted by the American Society of Nephrology, all rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare provider if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast of the American Society of Nephrology.